as we call it. Just gonna say, got it. Sorry, I'm just trying to get that off. That got it off my screen, which won't let me to. Let's see.
I apologize.
No worries.
Thank you.
Oops. I think when my thing is on laser, it's not letting me. There we go. Perfect. Okay. All right. And so the CTEP, which is the Cancer Therapy Evaluation Program. So this is the organization that is really in charge of gynecological cancers, amongst others. And when we submit trials through the NCI, this is who the initial group that evaluates them are for. And so you can see their mission here, and ultimately they're tasked with a collaboration of a research community and pharmaceutical and biotechnology industry to bring trials and other therapies to patients. And so as you can expect, there's the natural bureaucracy of going through government, and that's can be beneficial and harmful. Beneficial in that we get agents that typically wouldn't be available to people outside of industry. But detriments is sometimes these take longer to open up trials compared to running a trial through industry or investigator initiated. Another group that is near and dear to my heart is called the ETCTN, and this is a bit more of a selective group. And this is the Experimental Therapeutic Clinical Trials Network. So this network is designed specifically to look at early phase clinical trials. And I'll explain in later slides about what early phase clinical trials are, but I'm more thinking phase 1 and phase 2. And these are smaller number of patient trials, and they're looking more for investigatory drugs. And they allow us to investigate certain therapies that maybe a company isn't a large company like ones you've heard of, like Merck or something like that, or AstraZeneca, GSK, the really big pharmaceutical companies across the country. Smaller companies or even the larger industries to run and help supply drug for trials that we want to kind of investigate a theory or a pipeline of stuff that's been done preclinically. Now, I'll give you that there's a lot of rigor to get trials into this. It means that preclinical data has to be very substantive to be able to get to a trial at this standpoint. So it's not a willy-nilly type thing. If you end up on an ECTN type trial, this is not a trial that somebody just thought of an idea and hey, let's see if this works. There's quite an amount of investigation done prior to any therapy ever even reaching a patient. And then the NRG is a large national consortium that we regularly are part of and enroll trials to. This is also through the NCI. So the NRG is a group that brings together GYN oncologists, radiation oncologists, and other cancer specialists, and they help formulate which trials our field really want to bring forward to the NCI. And these can be larger trials, what we call phase 3 trials. And again, I'll explain that in a minute, through this consortium. So the second group of people who run clinical trials are industry. So industry themselves will develop a product or an idea, and then just like any other business, they want to kind of bring it to patients. And so they will run their own trials, and they'll host a number of sites, usually across the country and oftentimes internationally as well. And they'll bring it to us as investigators and say, would you like to open this up at your institution? And then those trials are run predominantly by that industry group. And so by that pharmaceutical company. And when that happens, they kind of have control of the data and where they go from that. But that helps them delineate to what population they want to try and bring these products to. And so this is much like, when I say product, these are therapies that we're talking about. The therapies that we're all familiar with are PARP inhibitors or immunotherapies or chemotherapy agents. And now the more novel therapies are coming through this way. And so this is more the collaboration of industry and investigators to help bring these trials. The benefit of industry trials is they're usually run pretty quickly and efficiently, because there is an underlying aspect that there's a business there. And so the industry is interested in running them more efficiently. And so, so that naturally occurs. And then the last is investigators. We call these IITs for shorts. And investigators are people like myself. So if I have an idea, whether I have a lab work or just an idea, as long as I have a scientific support for that, I can introduce my idea to a pharmaceutical company or the government, NCI, CTAP, whoever I think is most appropriate, basically to fund the trial. However, while the industry will, the pharmaceutical company will fund the trial, the investigator themselves is really the sponsor of the trial. And what that means is that I'll create a scenario here where I write a protocol or a trial called an IIT. I submit it to the industry, and then they say whether it's aligned with their goals or not, and they decide to fund it or not. But it's my trial. I'm the one who owns the information, the data. I'm in charge of making sure it's compliant with the FDA and everything else. But it's important that they know that the investigator is the one in control there. Now, they obviously have input and say. It's a collaborative effort, but the investigator is really the one with control over there. And then finally, there are collaborations between the two. So we are members of something called the GOG Foundation, the Gynecologic Oncology Group. And this is really an industry that's meant to and designed to bring together investigators and pharmaceutical companies. And they do a phenomenal job at this. And what they do is really bring us all together and try and streamline all of the inefficiencies. So they handle a lot of the contracting and the behind the scenes work for us to collaborate and bring trials to patients faster and more efficiently, and have the results out there faster too. So what we don't want to do is spend decades developing a drug, and that by the time the trial is done, the science and everything has already surpassed that. And this is an organization that helps expedite that in a very efficient way. So that leads to a lot of questions that I got. And this is a slide I added this year because it's a common question I get is, well, who pays for the clinical trials? And what I mean that is, ultimately, I often get asked the question is, how much am I going to be responsible for as the patient? And the short answer is, as a patient, you are not allowed to pay extra costs. So I say it's not free, because you still have to pay whatever your insurance has determined as standard of care, meaning that it would be the same as if you were undergoing any standard chemotherapy or anything like that from your provider, that whatever copays you have or aspects that related to your insurance that you have to pay, that is still applicable. However, anything that's related to the trial itself, you're not responsible for. The trial sponsor will pick up. And so you have no extra costs, but it's also not free to you completely. So anything that you would be getting on standard or pay for, like labs or things like that, you'd still have to pay for in clinical trial. This is actually a national law. The ACA made this a federal law that no insurance company can deny you the ability to go on clinical trial for cost. And these are the exact wordings that they use. So ultimately, clinical trial is available to you as long as you have the insurance and ability to pay for your standard of care. Now, that also becomes into a number of other things. A number of institutions will supplement that with other issues if you're uninsured, but that's getting a little bit into the weeds. So let's talk more about the clinical trial phases that we were mentioning before. And the phases of clinical trials really range from phase 0 to phase 4. Now, phase 0 and phase 4, I'm going to only talk about briefly because we don't have a large role in those as far as development of clinical trials. Now, phase 0 are really those initial initial studies that are looking at blood markers or tumor markers, and really just looking for validity of what the drug does to the body. There's generally considered no clinical benefit to the patient for a phase 0 trial. It's really investigatory. So oftentimes in our clinical portfolios here, we don't typically have phase 0 trials available here. Phase 4 trials are the exact opposite end of the spectrum. The therapy is usually already well approved by the FDA. It's already significantly in use. And now they're testing long-term safety in a larger, diverse patient population. These are usually thousands of patients. And oftentimes these trials aren't even done because once the drug is approved and in use, the safety data is already built into the previous trials, and trials like this may not be necessary. So the important ones to talk about are phase 1, 2, and 3. And so phase 1 trials are typically lower number of patients, and they are considered for safety only. Now, I say that with a little bit of an asterisk behind it, because while we're interested in the safety analysis of phase 1 trials, there's clearly an impetus to look for signals that this is working in certain patient populations. Historically, phase 1 trials were given to all patients with cancer, meaning that it didn't matter what type of cancer you had, you were enrolled in the phase 1 trial. We call those all-comers trials, meaning it doesn't matter if you have lung cancer, ovarian cancer, testicular cancer. It means that you could enroll in that same trial. Now, in our modern era of phases of trials, oftentimes even phase 1 trials have specific patient populations in mind. So I'll often have phase 1 trials that are specific for ovarian cancer or specific for certain types of ovarian cancer. And that's because the preclinical work has really elucidated that the signal for that therapy is patient specific or tumor specific. And so that's important to discuss with your provider when they're offering this. Now, once that signal of safety has been proven, then we look for the signal of efficacy, which has already shown something in the phase 1 trial. And so phase 2 trials are then a larger number of patient population, usually in the hundreds. And this is now looking to statistically prove that therapy is effective. And what that is literally usually just an expansion of the phase 1 trial. Sometimes there's some tweaks to it, but an expansion to incorporate more patients to be able to get the number of patients necessary to statistically prove that. And then phase 3 trials are when you have proven that that drug or therapy is efficacious, and now you want to confirm it to a large population and oftentimes compare it to the standard of care. So if it's maybe a comparison for versus a chemotherapy or versus something else, but you're trying to prove that it is equal or better than the therapies that are already available to patients. So one thing about trial design is that, as you can imagine, all of these take time. We talked about phase 1 through phase 3. And that's how you get the drugs approved through the FDA. However, each of these takes a significant amount of time, and with time comes money. And so historically, the drug development aspect would take decades, and 10 to 12 years was an easily considered a feasible time frame for a drug to be developed into readily available to patients. Now, this has been expedited over the past decade. The trial design is really now incorporated to better accommodate a faster development into patients actually seeing these outside of clinical trial. The reason that I believe this is because our science is vastly improved. We've learned a lot about biomarkers and about patient-specific care and tumor genetics and genomics and things like that, that the science is moving so fast that we really need these drugs developed faster. And so we do this in a number of different ways that, again, I'm not going to get into too much detail today, but there's a number of ways to expedite trial design in order to get these therapies to people faster, while not hindering any kind of safety aspect of that. So let's dive a little bit further into trial design, because I think this is important when someone is considering to be in a phase 1, phase 2, or phase 3 trial. Now, for those of you who have met me before and have heard my little spiel when I'm talking to you about clinical trial, I'm always going to recommend a phase 3 trial, then a phase 2, then a phase 1 trial. Sometimes there's some discrepancy in that, and I'll describe it to you specifically related to your tumor. But the rationale is that these trials are further along in development. And so it makes more sense for me to offer you a phase 3 trial before a phase 1 trial. But if a phase 1 trial is all that's available to you, it's important to understand how this works. Because I've told you previously that these trials are designed for safety. But whenever I'm talking to you about a phase 1 trial, the goal that I have with you as a provider is that we're going for efficacy, because I think that this trial is going to work for you. But these trials are designed in a manner to make sure that we're not endangering people, but without the inherent risks that you can imagine in any clinical trial. So from this aspect, this is the most classic way that we've designed phase 1 clinical trials. It's called a 3x3 design. And what it means is that into a specific design trial, we are going to incorporate a certain, if you can imagine, the most tangible thing that we can adjust is the dose of the medication. So if we start out at a certain dose, we're going to dose 3 people with that same amount. And then we've already delineated what the toxicities we can expect are, or unforeseen toxicities. And from the toxicities that people experience, that determines whether we move forward with increased dosing. And so here's an example that if no patients experience any toxicity, then you move on to the next higher dose. If one person does, then you enter 3 more patients into that dose to get more patients and see about the number of toxicities. And then if 2 to 3 patients experience a certain toxicity, then that's considered the maximum tolerated dose, or the MTD. And then that's determined whether we think it's efficacious or not. Because in a phase 1 trial, we're looking for 2 things. One's called the MTD, the maximum tolerated dose, and 2 is called the RP2D, the recommended phase 2 dosing. And those are oftentimes the same, but not always. And so this design helps us determine what dosing we should recommend for further expansion of this therapy. Now, there are a number of different designs. I just mentioned that the 3x3 is the classic one, but there's other things called the accelerated titration design, and there's literally many examples of how to do this. In the accelerated titration design, we give one patient a certain dose, and then we give them, if they tolerate that dose, we give them a higher dose at the next round. And if they tolerate that, then a higher dose. And we do that until there's a toxicity. And so, and then once there's a toxicity, we introduce more patients into the population getting that therapy and figure out if that toxicity was limited to that one person or more people. So as you can imagine, there's risks and benefits of each of these designs. And I use these 2 as just examples, but there's many more. But we're trying to minimize the amount of toxicity to people, obviously, but also learning to make sure that we find a dose that's efficacious and we don't run the risk of getting a drug that at too low of a dose that may be truly an effective drug, but we never got it to enough dose that it is effective in people. So lots of things to weigh in balance here. So in hearing all this about safety, I think one of the natural questions is, well, why would I ever go into a phase 1 clinical trial? This sounds a bit scary, and where am I going? And the truth is that there's a lot known about the therapy before you ever even enter it. And this is some work that I did and published about ovarian cancer patients on clinical trial, on phase 1 clinical trial at the University of Colorado alone. We looked at the patient population who were going on clinical trial, and I found some interesting things. The first is that many patients went on more than one phase 1 clinical trial, which meant that the therapies were working for a time period and enough that the patients were healthy enough to continue to go on other clinical trials. And as you can see here on the left, while 60% of the patients had never been on a trial, over 30% of patients had already been on one, and we have several who have been on 4 or 5 clinical trials, phase 1 clinical trials. And then you can see here that the number of prior therapies that patients had are quite high. So somewhere up of 5 to 7, some even greater, kind of a standard bell curve here. And so the data over the past 2 decades have really demonstrated that there's a significant safety and good results of this. So, oops. So what we looked at is what were the responders of this. And what we showed is that when we talk about response, we talk about a couple different terms. One of them is called ORR, or objective response rate. And that means people who have had a partial response or a complete response to chemotherapy. Now, complete response, meaning that all of the tumor goes away, is uncommon at this stage of the game. But partial response can occur. And then what's very important is clinical benefit rate. For those of you who I've met before, you've heard me hear that at later stages of ovarian cancer, if we can get the tumors to stop growing and not cause symptoms from therapy, that's a real win. And we call that stable disease. And if we can keep disease stable without much toxicity for an extended period of time, in my book, that's a win. Obviously I want to cure the cancer and I want to get it away. But with the knowledge that we have now and the therapies that we have now, I do consider that a win. So seeing that 50% of people had what we call a clinical benefit rate, including stable disease, is significant, because as you saw in my previous slide, that patients had a number of lines of therapy before. And when we think about therapies working, we think about them working in the order of months, not years, when we're talking late stage therapies at that point. The other important thing we found here is this is a survival curve, and it's looking at what we call progression-free survival and overall survival. And the important thing to point here is that the progression-free survivals and overall survivals for this patient population, and what I mean by that is the heavily pretreated patients who have been on 5 to 7 lines of therapy or more, is quite congruent with any therapy that we know exists, even standard of care therapies, chemotherapies. And what this showed is that that the therapies that we're giving in phase 1 trials are safe and about as efficacious as other standard therapies. And the important thing to think about here is there in the 1980s and 90s, there was this big ethical concern. Are phase 1 clinical trials ethical to give to patients? Because there was this thought that, well, it just should be available for people who have no other options, and we may be causing more harm by introducing these agents and causing death. And the truth is that we're not. And so what we're showing is that these trials are safe. No patients in our decade of doing this died due to a toxicity of the therapy. And only 2 patients actually died on trial over the decade. And that was due to disease progression, not because of the trial itself. So let's move on to phase 2 trials then. So phase 2 trials, as I mentioned, are where we are comparing we're expanding the number of patients on a trial to see if it's statistically efficacious. To do this, we're also looking for a bit of a comparison. And when we do this, we have 2 ways to compare a therapy. We can do it compared to a placebo. So we can get an actual placebo drug and say, is one better than the other? And the other is compared to a historical control. So if we know that a certain patient population, let's just say, high-grade serous ovarian cancer with X number of lines of therapy, we can say statistically this patient population has a progression-free survival of X amount of months. We can compare it to that and see if it's better, the worse, or the same. Phase 2 trials don't always have a placebo control, so it's important to talk to your provider. Does this trial have a placebo in it or not? And what are the risks of having a placebo or not? Oftentimes, these are what we call single-arm trials, and they have just the therapy, so we know exactly what you're getting, and we know that there is no placebo. Just an important point to talk about with your provider when talking about clinical trial. And then phase 3 trial design. So phase 3 are, as I mentioned, these are going to be the large thousands of patients across the country or world, and looking to see is it compared to standard of care. Now, this is where we get statistically kind of interesting, because there are ways to design a trial that can either hinder or assist your rate of having a positive trial, meaning that we get this therapy to patients. And there's three main statistical designs of this, and that's superiority, non-inferiority, and equivalence. And what this is looking at is, how do you statistically design the trial? And do you want to prove that your therapy is better than an existing therapy? Or do you want to prove that it's equivalent, meaning meaning that the two are not too different, or that it's not inferior, meaning that it's a little bit different than equivalent, but it's generally thought about in that same standpoint. We often use non-inferiority to prove that if your belief is that your therapy works just as good as an existing therapy, but maybe way less toxic, then you're more likely to have your therapy prescribed. And so a non-inferiority trial may be the best way to do that. The interesting part that comes of this, though, is that if you design a trial for one versus the other, the biggest difference is the number of patients you need. And so as you can think about this, if you have more patients in a trial, it costs more and also takes longer time. So you may not get your results as quickly. But certain trial designs require more patients or less patients. And that factors into how you design that trial and whether that trial is going to get out quickly or not. That is just one of the things that we think about when we're designing a trial. Other things are eligibility. And so eligibility vastly affects who comes into your trial. So one of the biggest things about clinical trials we talk about with anybody is the screening period. There's oftentimes a screening period where we have to check imaging or labs and make sure you're a healthy enough patient to enroll on a clinical trial. The reason why I mean that is that if you fill a clinical trial with patients who are not healthy enough to receive the therapy, then obviously you're going to fail the trial, where it may actually be an efficacious agent, but just not that we were finding out that it wasn't the agent causing harm, it was the patient's medical comorbidity. So if someone had a significant heart problems, and this drug could affect that, we'd want to know that, but we would want to make sure that that's not conflicting data. So this gets into what we call the inclusion-exclusion criteria. And this is where we kind of really go into validity versus feasibility. And validity is, validity and feasibility. So if we incorporate too much patients that if we allow everybody into the trial, then we suffer on our validity, because that ultimately means that we haven't really narrowed down who this works or doesn't work for. But if you have too narrow of a focus and you don't allow enough patients in, then you really limit your feasibility, meaning that you can't actually enroll enough patients onto the trial to make sure that this therapy is feasible. So it's finding that right balance of validity and feasibility, of finding the right patient population. And then there's randomization. So randomization is a way to remove bias. And for some trials, not all trials, we get randomized to different arms. So if we're comparing it to standard chemo options, you may get randomized to the trial therapy versus a standard chemo option. There may be mixtures of those in there. And sometimes we know what you're randomized to, and sometimes we don't know what you're randomized to. And this is why that kind of gold standard is what we call a randomized placebo-controlled trial. It means that the providers are blinded to it, the patients are blinded to it, and we're really removing some of those biases that we each have and allowing really the study data to dictate whether this is an efficacious therapy or not. That allows us to eliminate these imbalances if we kind of remove as much as we can, because we all have inherent biases. I may have a bias of which trial I offer you. The inclusion-exclusion criteria may have a bias of focusing more towards one patient population versus another. You may have a bias as far as about therapies you want to go on to versus not. We all have inherent biases, and it's impossible to remove all of them, but randomization allows us to minimize that as much as possible. All right, and so biomarkers. So biomarkers are pretty much mandatory in most clinical trials these days. And these can range from a number of different things. Sometimes we're exploring biomarkers, meaning we're trying to find out if there's a certain biomarker that a patient has, or a tumor has, or the blood has, that makes this therapy more efficacious or less efficacious. And so there's kind of 5 main categories of biomarkers. There's prognostic, predictive, integral, integrated, and exploratory. And these are built into clinical trial, and sometimes they're mandatory and sometimes they're not. And that's what we mean by integral, integrated. So as an example, prognostic tumor markers are things like tumor stage, grade, or surgical outcome. These are things that are just kind of facts about a patient's care, and we're looking to see if they work more in stage 3 versus stage 1 cancers. Predictive would be a response to something. So let's make an example of an estrogen receptor or progesterone receptor. We predict that a therapy is going to be more responsive to estrogen receptor tumors. Integral is that it's integral for you to be in that trial, meaning that you have to have a certain biomarker to be enrolled in a trial. An example would be, let's say there's a PARP inhibitor therapy trial that they're only allowing patients with BRCA1 mutation. So that would be an integral biomarker for that. Not all trials have integral biomarkers, but some do. And then integrated is testing to see if that biomarker is actually part of who works. So it's the kind of opposite of that, is allowing all patients in, but then evaluating whether BRCA1 patients work better or worse. And then exploratory is we're looking at new ideas, and it's just really to help answer new questions. So this would be, blood biomarkers, tumor biomarkers, across the board, a number of ideas that this could be for the clinical trial. And these biomarkers are really what's shaping clinical design these days. And so here's 2 big kind of trial design options that are really biomarker specific. So there's things called umbrella trials, and this is based on a specific tumor type that then, based on the biomarker, you get different therapies. So this example you have 3 patients with lung cancer, but they have 3 different mutations in their lung cancer. And so based on those 3 mutations, they get 3 different drugs. And so does one patient do better than the other? The opposite of that is a basket trial. It means that you can have any cancer type, but one specific biomarker. So an example here would be that each of these 4 tumor types, lung, liver, stomach, colon, each of these has a certain genetic mutation, and that based on that genetic mutation, we give one therapy. And is that therapy efficacious in that mutation? And so this is how biomarkers can be incorporated into clinical trials these days. Next, let's talk about how we assess people on trial. The most common way we do this are 2 major factors. One is the therapy working? And we do this by imaging. We call this RECIST evaluation. And that's an evaluation of objective response rate, an objective way to look at the imaging to see if tumors are growing or shrinking or staying the same. And then adverse events. This is solely based on toxicity, meaning that we grade all toxicities. It doesn't matter if you get a hangnail on this therapy, we want to know about it. And then we kind of determine, is it related to it? Is it not related to it? Is it possibly related to it? So let's dive into this a little bit. So when we look at the imaging, for those of you who have been on clinical trial or are thinking about clinical trial, we get regular imaging on pretty much any clinical trial. And the rate of that, whether it's every 2 months, 3 months, kind of depends on the trial design. But this is the kind of thing that we're looking at, is RECIST measurable disease. And these are objective ways that we mark out how your tumors are doing. And we measure certain lesions on the CAT scans and then evaluate them. And we have certain numbers that we can evaluate. We have certain numbers from different locations in the body that we can evaluate. And this can get into the nitty-gritty. But when we look at them, we're looking for 4 major responses. And this is one of the things I was alluding to before. We call them complete response, partial response, progressive disease, or stable disease. And this is based on whether the tumors are growing or not. So you may hear your provider talk about how the disease is growing a little bit, but you're still considered stable disease. And that to me is a win and still able to stay on trial. And so we look for that duration of how long we keep patients on and they're responding to either of these. Then it gets into a little bit more nuance of target lesions and non-target lesions. And then if it's an immunotherapy, we can get into the weeds of how immune therapy response affects tumor growth. Sometimes they swell and they shrink. But let's get a little bit more into adverse events and toxicities, because these are obviously quite important as well. As I said before, we grade everything. We grade whether your blood pressure was a little bit high or a lot high, and is that related to therapy or not? And the way we do that is by grading things from a term 1 through 5, and everything can be graded. As I said, as little as a hangnail or as severe as blood values. The blood value ones are the easiest to be objective to. As you can imagine, we come up with set criteria, as you can see here on the bottom, of anemia. And there's set numbers, and based on what your numbers are, we grade it. But then we help determine, is this related to therapy, or is this just related to something else in life, from the disease or from some other medical comorbidity that the person may have. And these all range from 1 to 5. In general, the terms 1 basically mean not clinically significant in any way, ranging all the way to 5, which is death. So the other thing that we look at when we're designing trials are trial endpoints. And this can be a host of different things, looking at response rates, the time to progression, because as you can imagine, if a therapy is effective, that's great, but we also want it to be effective for a prolonged amount of time. So it matters whether a therapy is effective for a year or 1 month, because that matters for prognosis. And then the most common that we use are progression-free survival. So the time of patient entering onto a trial to the time that the disease progresses. And then overall survival is time to death. And then there's patient-reported outcomes. We often have questionnaires looking at how tolerable is this therapy. These are oftentimes incorporated. And when we do this, we're doing this for a reason. We have different endpoints of a trial because the therapy, our ultimate goal is to get this approved by the FDA and get it to patients without having to be on trial. The FDA has delineated certain things that they will accept for FDA approval. And if the endpoint is overall survival, it's kind of unanimous that if you improve overall survival, this drug will get approved. But it's not as easy as that. They oftentimes need multiple trial endpoints to incorporate whether the FDA will approve a drug. And so in conclusion, I really wanted to talk about that. My opinion is that clinical trials improve our knowledge about treatments. They improve options for patients, and they improve outcomes for patients. Oftentimes trials can be a scary thing because people don't want to feel like a guinea pig or a lab rat. You know, I've heard those terms oftentimes before, but hopefully what I've shown you today is that clinical trial is really an exceptional option for a number of people, and that I'm the first one to tell you if I think that a trial is not the right thing for you or if it is the right thing for you. You know, people ask me, well, is this trial going to be better than the standard therapy that was offered to me by my other provider? If I knew that, then we definitively say this is the right thing for you. That is the whole reason that we're doing trial design. But I do think that trial design and trials now are oftentimes access to the latest and greatest and have significant potential. The other thing about trial design is that we are purposely keeping a very close eye on how you and the cancer is doing. And when we do that, we allow for you to come off trial easily and quickly so that if it's not working, we move on to a next therapy that hopefully will be more efficacious. But I do think that they're a great option for many people. The resources about clinical trial, there's two big ones. The first one, absolutely just talk to your provider. Say, hey, have you thought about clinical trial for me? Am I at the right time frame for this? Is this something I should be thinking about? And then the other is doing your own homework. Clinicaltrials.gov. This is an open access website. They have pretty simple terms that you can enter in there. You can say, I have, ovarian cancer, and then it'll also list the locations that trials are available, because trials aren't available everywhere across the country at all sites. Certain sites pick up some, certain sites pick up others. But ultimately, these are kind of your biggest resources to evaluate for clinical trial. And then I definitely wanted to give time for questions. So I really just want to thank Coca again for allowing me the opportunity to give this talk, and I'm happy to entertain questions from anybody.
Thank you so much, Dr. Corr. That is, for something that's opaque, it's really great to have some pieces of that. I just want to add a couple of, I want to summarize and see if I got it right for everybody, and then we can move on. So number one, I also want to add that the Clarity, as in Clear Eyes Foundation, has a trial finder specifically for ovarian cancer, which is another option besides what Dr. Corr listed. And then I just want to reiterate that you're saying that part of the national guidelines is that people should be offered clinical trials if they have ovarian cancer, correct?
If it's available to them, because one of the things that the NCCN recognizes is that the reality of the just geographic dispersity of our country is that someone in rural Montana may not have the accessibility to get a clinical trial, meaning that they may not have the financial ability to travel to a cancer center trials network area or site. And so that stipulation of if available to them is pretty important in my mind, because sometimes a barrier to clinical trial is just simply financial burden and being able to travel to a site. So yes, it should be offered to you if available.
Can you remind me about the clinical trials? I'm guessing should not be seen as the last stop when nothing else is working. I mean, what's the framework again? If you could repeat.
So I'm a very big proponent of that. That's actually was the impetus that trial that I told you that we published a few years ago. The whole idea of that for me was to tell providers and patients that clinical trial, including phase 1 clinical trial, should be considered at every stage along the game. Now, a phase 1 trial in an upfront therapy is not appropriate. They also don't really exist. But that at any time of recurrence, clinical trial should be considered if it's feasible for you. And the main point I want to say is that clinical trial is not a last effort, and that if your provider's saying, well, it's either go to hospice or try clinical trial, I'll absolutely talk to you about your clinical trial options, but that may not be the best appropriate time.
Yeah, I guess because there is, I think, this population is probably more educated, but I feel like kind of just lay people in general view it that way. And I think that people diagnosed with ovarian cancer need to think of this as like another tool for you, and not that option at all of last resort. That, again, when you looked at that chart that you had, Dr. Corr, it was really encouraging to see how well people did, and that it did not leave them in worse shape at all. So that was, I think, pretty exciting for that. Well, listen, it's hard to ask questions on this, so I think we'll just be polite to one another. You can just unmute and speak up, and if we have more than one person, we'll just have a little bit of a line, a little bit of a queue.
I have a question.
Go ahead.
So if I'm at Anschutz, and if Anschutz does not have a trial for me, then if I say go to MD Anderson, does that mean that I stop receiving all of my treatment at Anschutz and I basically transfer to MD Anderson or some other major hospital that has clinical trials?
Great
How does
question. The answer is sort of, and let me explain what that means. So I'm always a big proponent of getting more opinions and seeing what options are available to you at different sites, and whether that's, you know, across the street from us over at Sarah Cannon or down at MD Anderson. One of the aspects of clinical trial is that you can't enroll in a clinical trial at MD Anderson and get your care here at Anschutz. And I'm using broad spectrums of our specifics of our location. But as your provider, you would not lose sight of me being your provider. Again, I'm just using generalities of me. Let's say I was your provider. It would mean that during the care of that clinical trial, you would be getting your care at MD Anderson. You'd have to physically be there. You'd have to get the labs with their delineation, whether they can be done at MD Anderson. Sometimes they allow local labs, but you would have to travel to their site for treatment aspects. Your provider at Anschutz wouldn't and shouldn't lose sight of you. Some providers choose to check in with you regularly at certain intervals. Some say, hey, you're going to be going under a lot of, you know, travel and everything. Why don't you not see me during this time? Just send me some notes, or, you know, I'll see you when this trial is over. That's kind of up to the relationship you have with your provider about how much to check in. But your provider can't prescribe the medication of that trial at Anschutz.
Thank you.
I have a comment and a question, if I can go.
Sure.
This is Jean. Yeah. Hi, Dr. Corr. It's good to see you.
Good to see you too.
Yeah. And I was in a trial with Dr. Corr last year, antibody drug conjugate. And I just want to encourage people to consider trials. Mine was very successful. And it's an opportunity, you know, I used to think of it as sort of a last ditch, but an opportunity to take advantage of cutting edge treatments. And then you have standard of care to fall back on, of course, always. And then also the reward of participating in the science and you know, contributing to what's going to be available for ovarian cancer patients. There's that too. I do have a question. I was curious about trial regulations around releasing data as that accumulates, and wonder about that.
Yeah, so the data during clinical trial is never released. The main reason that is actually a statistical thing, is that even doesn't matter who's running it, the NCI, the government, you know, the industry, the investigator. Every time you dip in to look at the data, whether it's working or not, it takes out something that we call alpha from the statistical design, and that makes it harder and harder to actually prove that the therapy is working. It's a statistical process that I still have trouble wrapping my head around. But basically, every time you go look at the data, you have to increase the number of patients for it to statistically be proven that it's working or not working. So they oftentimes delay it until they power an analysis by saying, you know, in order to prove the point that we want to prove, we need 600 patients. And they're going to wait till they see that 600 patients. Now, there may be things designed into that they're going to say, well, at 100 patients, we're going to look at it to make sure that we're not doing harm, and then go on to the 600. But that'll be built into the statistical analysis. So the short answer about during trial, none of us will have the results. After trial, after complete enrollment, then there becomes that time period of, well, we need to wait for events. And so what I mean by events is, if we were looking at progression-free survival, we need a certain number of patients to have progressed for us to be able to get any results of this. If they were looking at overall survival, unfortunately, we have to wait for people to pass away to get that data about. That's why usually progression-free data comes out before overall survival data. And so that data, trust me that everybody involved wants to get that data out as quickly as possible, because that's what helps move things forward. So oftentimes it's first presented at a meeting. So we have another national, international meetings. Oftentimes that's the first presentation. And then secondly, the publication usually follows that pretty shortly, depending on how big the data. Sometimes it's congruent, the data, they'll be waiting publication and it'll go out the day after it was presented. And sometimes it's not as important and clinically relevant. And so it takes a couple of months to come out. And then it's in the public domain and available to everybody.
Thank you.
The short answer is the data, then the trial you are out is not 100% out yet.
Right.
I know why you are asking.
Yeah. I was able to see it very early.
Yeah.
Phase 1.
What else can I answer?
I have a question. I'm Jean, and I would like to know why you have mostly phase 1 trials and why not more phase 2 and phase 3?
Sure. We do have all types of phase 1, 2, and 3 trials available at any given time. Our research portfolio is usually always in shift, and that always depends on, one, the trials that are available nationally, and two, it depends on when trials open and close. So oftentimes phase 3 trials actually, while they open fast, they also close fast because they enroll nationally quite fast. And so those are always in shift. The other aspect about clinical trial portfolios is that to open up any trial takes a fair amount of time. Oftentimes it takes about 6 months to open up a trial at an institution. We strive to open up a trial within 3 months, but because of a number of different things, it can extend that to upwards of 6 months. That means I do my best to have the foresight to keep trials open and all the time. But our goal is to have several trials available to all tumor types across all gynecological cancers. So while at some point certain phase 3 trials may not be available to certain people, always check back in with your provider to see if anything is coming and down the pipeline or when things are be opening. But we always try to have multiple trials open.
Very good, thank you. And one more quick question, and this is, I had read about umbrella and basket trials a while back ago. What percentage of trials now would you say would be that one of those 2 kinds?
Only a few, and the main reason is because a lot of these trials for basket or umbrella trials are usually large trials run by the NCI or government. So there's a large national trial called the MATCH trial. So that's looking at a bunch of different tumor types, and that's one is more of a umbrella type. But the greater focus for kind of disease specific end up being more that inclusion, like a specific biomarker, gets built into the trial design itself, but doesn't actually become an umbrella trial because they don't want to incorporate too many different types of tumor types or things like that.
That makes sense.
Sorry, I had to remute a few of you because somebody's typing was getting in the background. So if you were unmuting because you had a question, please unmute again. Diane, I see you unmuted.
I have a question.
Hmm?
If a patient did have the financial resources to go to a distant location, would you recommend it?
Depends on the trial. I've often had patients who say, I'm meeting with you, Dr. Corr, today to see what you have available. And in full disclosure, I'm meeting with MD Anderson on Wednesday. I'm encouraging you to absolutely go there, see what they have available and what they offer you. And then I'm always available to compare and contrast. My goal is not to get you on a trial at my institution. My goal is to get you the best care that is right for you. And so I oftentimes will get messages and say, hey, Dr. Corr, MD Anderson or Mayo Clinic offered me these 2 trials. Would you mind taking a look at them? And I'll say, hey, I've looked at this one, I understand why they sent it to you. I think this is a great option for you, or no, I think that this other option might be better for you. And that's, my opinion. But that's kind of, why you came to me for my opinion. So I'm one to kind of say, I think looking around is a good idea. The only caveat I will say is where I found harm is in when people shop a little bit too much. And what I mean by that is I've had some people say they want to go to me, Sarah Cannon, MD Anderson, Mayo, Memorial Sloan Kettering, and they spend time, effort, money going to 6 or 7 different locations. And the therapies being offered end up not being too different. But what you have sacrificed in that is time off of therapy. And that's where I worry about it and say, hey, absolutely seek as many opinions as you can, as you should, but be mindful of the time it's taking and that time that you're off of therapy. And is that the right thing for you?
Can I have a quick question for you. If, should all of the providers, all gynecologists be able to make that same assessment that you make? Or is it because you're in this field more that you have a different, maybe an elevated sense of what's going on out there?
Yeah, great question. I can certainly talk to my partners. I can't talk to all of the GYN oncologists, across the country and stuff, because that obviously, like anything, there's a certain level of knowledge that is variant. I would feel comfortable with any of my partners, bringing this information to any of my partners and saying, hey, what do you think about this? Some of them might overtly say, hey, why don't you talk to Dr. Corr or Dr. Babak or, someone like that. And some of them may say, no, I'm comfortable answering this. But I would feel comfortable that any of my partners would be a great resource, that you do not have to speak to me directly. You can speak to my partners.
And this is Jean once more, just very quickly. In 2008, my husband and I went down to MD Anderson for a second opinion, and they asked me if I would be interested in a clinical trial phase 2 with docetaxel and aflibercept. And I said yes, and I will tell you that it ended up with my flying down on Tuesday, having chemo on Wednesday, coming back on Thursday. And I did that for 9 times, but it was a complete response. So it's quite well worth it. But you do have a hotel place to stay, you have food to buy. And so it ended up for us being a little expensive, but of course, well worth it. How can you put, money against your health? So it did work out for us. And so as the other Jean says, really look into clinical trials because they can be wonderful.
And that's a prime example of why I would say, absolutely go shop around. If MD Anderson has the best trial for you, then by all means.
But this is just my own ignorance. There are some trials, I would imagine, are you both enrolling people in?
Yes, absolutely. And so that's why I encourage you, if you do get a second opinion, ask if we have that trial open, and then ask why, we didn't offer it to you, which I think would be an extremely rare scenario. But yes, if we are open and MD Anderson is open, and I'm again just using MD Anderson, but we would absolutely be able to be your provider if we have it open here.
Yeah, I guess I was just thinking about some of these larger trials, like you said, have enroll across the country, so you don't necessarily have to travel to another location.
Correct. Yeah, and the clinicaltrials.gov will be a good resource for that. It'll tell you what sites are open for that trial. It's not always 100% accurate, but it's pretty close.
MD Anderson has the, oh, I'm sorry.
No, please go ahead.
Oh, I was just going to say real quick, MD Anderson has a branch at Banner Health in Greeley, and I don't know if it's possible with trials, that you hear about in Houston or whatever to then receive them at Banner Health.
That's getting a little bit logistics of contracting, and the short answer is I don't think so. Unfortunately, sometimes there are, sometimes the use of the MD Anderson label is sometimes more of a monetary thing than an actual provider thing. And sometimes it's not. I'm not as familiar with that site. And so kind of depends on what they're contracted to be able to do and which providers are there.
It's a good question to clarify for us. Thanks for asking it, Jean. Well, listen, I hope everybody got the message that it's another resource that you should investigate, and it's worth bringing up with your provider. And like Dr. Corr said, if for some reason you're not getting offered those, to ask, or it's always second opinions are always just more information. And we always encourage you to be a good advocate for yourself. I'm so grateful, Dr. Corr, for your time. Thank you for
Thank you.
elevating this for all of us. Sure.
Thank you.
Thank you again. And good
Thanks
to
so
see your faces again.
Yay. Okay. Well, we'll hopefully see you all soon.
Thank you.
Thank you.